Professor Tanjiro Koppelman from UCSF.
Okay, thank you. So you'll find some of the themes that Hua talked about echoed in my talk as well. So what I was planning to do is to talk about two aspects of our work. In the first part, we're going to look at how we're trying to think about foundational methods, computational methods to design molecular parts, where we are right now with computational design, and how we're trying to learn how to improve our methods. And then in the second part, I'm going to talk about a specific application applying computational design to re-engineer sets of protein-protein interactions, both in vitro, as well as some initial experiments to transfect these engineered proteins into mammalian cells and test for phenotypic output. Before I start, I just wanted to highlight the people who did the work. The first part of the computational method development was done by two really terrific graduate students in the lab, Elizabeth Humphries and Dan Mendel. We had help in a very nice collaboration with an applied mathematician, Miguel Icuzias, at the University of Mexico, who helped us with some of the algorithmic development. And then the second part, the experimental work on re-engineering sets of protein-protein interactions, was done in my lab almost exclusively by postdoc Greg Kapp. But again, this was a terrific collaboration with Wendell Lim's lab, and I think Wendell is going to touch upon sort of the larger aspects of this collaboration in his talk tomorrow. So foundational methods for molecular design, ideally, where would we like to be? Well, we'd like to have predictive computational methods where we can input some kind of mathematical description of fitness of our parts, and we would like to arrive with a sequence in a molecular part that obeys those fitness criteria. And we like, as I said, for this procedure to be predictive. Of course, we're not there yet, but these are the basic big picture goals, and perhaps you keep them in mind while we're going through what are the kinds of improvements are that we're trying to make to computational protein design methods that I'm going to mention in the talk. So we had some introduction on computational protein design already. I'm just going to go very briefly through the method that we are developing and applying. This, of course, builds on real pioneering work of a number of different people in the field, and some of those are listed up here. And so what I'm going to talk about is our particular incarnation of computational protein design, where we develop and apply the Rosetta molecular modeling package. So as input to computational design, we start generally with high-resolution structural information on either isolated proteins or protein-protein complexes or protein-ligand complexes, protein-DNA complexes, for example. We have a certain design objective, and I'm going to present a couple of examples for what these design objectives could be. And then as an output, generally what we like to get is one or a few low-energy sequences that we think, according to our methodology, are consistent with both the structural constraints that are placed on the problem as well as our design objective. So then the general methodology that is in common for many of these computational protein design methods is that the side chains, at least in the area of interest, for example, a protein-protein interface, so the interaction area that we'd like to re-engineer, the side chains are removed in that area. And then most commonly, protein design proceeds using a fixed polypeptide backbone, although I'm going to go into a couple of improvements that we've made recently to try to move away from that approximation and allow for the naturally occurring flexibility of proteins and protein-protein interfaces to take that into account. Then for this area of interest, for example, a protein-protein interface, we can now explicitly represent all 20 amino acids. So we can generate large libraries of side chains computationally, and each of these amino acids is also represented in a variety of different conformations that we heard about. Generally, these combinations of sequences as well as conformations are too large to completely enumerate, so we need computational algorithms to search through this space. We heard about genetic algorithms, which is one of the methods that is commonly used, as well as Monte Carlo search techniques, which is the one that we use most often. And then at the heart of this process really is a scoring function that tells you which of these sequences obey your fitness function and which ones don't. We developed the Rosetta scoring function for this process. In general, very briefly, it's dominated by physical terms which describe the packing interactions between atoms. We have an explicit hydrogen bonding potential as an approximation of at least short-range electrostatic interactions. Long-range electrostatic interactions are a definite problem here. And we have a simplified implicit starvation model. That's one of the other major approximations here. And then as I said, we end up with one or a few low-energy sequences that we think, according to the scoring function, are consistent with our objective. And so one of the objectives that you already heard about is to design novel folds. And again, as you heard about, this has been done in David Baker's lab by Brian Kuhlmann a number of years ago. And so this is the backbone of TOP7, a protein fold that did not exist in nature. What I'm showing you here is just a simulation of looking at the protein core, trying to simulate all 20 amino acids at each of these positions in a variety of different conformations. And we end up with a prediction of this protein core that turns out to be very similar to the actual crystal structure that Brian Kuhlmann and Gautam Dantas in David Baker's lab solved of TOP7, showing good agreement with these initial design calculations, and actually generating a very stable protein with a novel fold. Another design objective that you can think about that we also heard about is you can think about engineering protein function or protein-protein interactions. Either de novo interactions or changing, for example, the affinity of protein-protein interactions, or something that I'm going to touch upon a little bit later, the selectivity of protein-protein interactions. So here's an example of something that we've worked on a number of years ago, where what we were trying to do is to take two protein domains in two distantly related proteins, where these two domains did not interact with each other, and try to see whether we can generate a novel protein-protein interface between those domains, and then characterize it structurally to make sure that our predictions were actually accurate. So I'm going to show you here just the end result of this. So this is work that my lab spent a number of years on developing this methodology. This was really one of the first examples of this, where here I'm showing you the agreement between the crystal structure of these two domains in yellow and blue here superimposed, and showing agreement with our initial prediction from computational design shown in gray. In this protein-protein interface, we designed 14 positions in total. This just gives you an impression of what the number of possible sequence conformations theoretically is in this interface. And of course, the structural space here is even larger, since we're representing everything at full atom detail and removing the side chains around. So you see good agreement here, forming a stable interface that we could crystallize and structurally characterize. One of the reasons why this, in fact, worked so well was that one of the major approximations that we're making, that we did make in the design of this protein-protein interface was true, which is that only the side chains that we changed in this interface, in total 8 mutations, only the side chains moved, but really the conformational changes at the backbone were very limited, and we did not actually model any backbone flexibility in the design of this. So one of the real next challenges is, since we know that proteins change their conformation upon binding interactions, can we take that somehow predictively into account to improve our computational methods? And so traditionally, or maybe I should go back, one of the other points that I wanted to make here is that most of these interactions here are also formed by elements of regular secondary structure, which are less likely to change their conformations. But this is vastly different for loops that are very important in recognition, specifically in protein-protein interfaces. So then Dan Mendel, in collaboration with Vegeli Koutsias, so Dan Mendel in my lab and Vegeli came up with a methodology to predict at high resolution the structures of protein loops, where essentially what this simulation is showing, and maybe I should start this up again. But the simulation is showing that they can fix the two endpoints of this protein loop, and then using algorithms from motion planning, look at or solve analytically the equations of different degrees of freedom of this protein loop that would still close these two endpoints here, and be consistent at the same time with the geometry of naturally occurring proteins. And you see that the end result of this is that the predicted loop, which is the lowest energy conformation that we obtained in the simulation, is actually extremely close to the crystal structure of this loop. And so if this worked predictively, I think this has enormous potential for reshaping the surfaces of proteins, specifically interacting with other proteins, to generate very, very highly selective new protein-protein interfaces, or in biosensor applications, for example. Of course, in order to really do that predictively, we need to show that we can not only do this in one case, but we can do this repeatedly and predictively. And so Dan and Vegeli then went on to test this out on a data set of 20 proteins, where we're looking at 12 residue loops, which are generally considered to be quite challenging, although, as you see, possible to predict. I'm just going to show you a collection here of examples where really, in more than half of the cases, you see an excellent agreement between the prediction and the crystal structure of these protein loops, although there are some examples where you do see deviations. But the bottom line here is, if you look at the median loop RMSD, so the agreement between prediction and experiment, you get in most of the cases in the data set, significantly below 1 angstrom RMSD, which means that this puts us into the range where we can actually start thinking about not only modeling these loop conformations, but changing their sequences at the same time and using this predictively for protein design. Also to show here on the same benchmark set, this new procedure here that Dan Vegeli came up with is a significant improvement over what we had previously in Rosetta, and it also favorably compares with what we found is a standard published state-of-the-art method, again, on exactly the same data set. So what I've shown you in terms of developing new methods for computational design, that we made some progress in improving methods, particularly taking into account backbone flexibility, at least in the structure prediction sense, not necessarily yet in the design sense. So you might wonder, in terms of using this now, applying it to synthetic biology, what I've just shown you requires an enormous amount of detail at high level, at the high atom level of detail. So how could this be useful to make biological engineering easier, where you don't have to, for each case, separately apply these extremely complicated procedures? And I'm going to argue that there are at least two aspects where I think this could make a contribution. So the first one, in terms of these very detailed simulations versus the abstraction that we'd really like to have in order to make biological engineering easier, is that this level of detail, the structural detail, gives us information on certain constraints in the system that I think we can use predictively in synthetic biology applications. So if you think about the notion of not just looking at, for example, this is a particular position in a protein that happens to be a threonine in the native conformation, well, maybe it can not only be a threonine, but it can tolerate a number of different other amino acid substitutions. And this is generally true for many proteins and protein-protein interfaces. So perhaps we can use this design methodology to predict families of not just single sequences, but tolerated sets of sequences, and then use this information to engineer parts families that we can screen for desired specifications. And secondly, where this detail of computational protein design might be useful or perhaps even essential is if you would like to use design proteins and design protein-protein interfaces to probably interface with existing cellular processes, where you'd like to, for example, engineer sets of proteins that specifically recognize other proteins to elicit biological output, or you might want to do the opposite, where you have your engineered set of interacting proteins, but you would like to insulate these from complicating other interactions that might occur in a cellular system. And so here you might be able to use computational design to look at the selective recognition of specific proteins while at the same time trying to eliminate some of the complex crosstalk to make these interactions orthogonal. And so, let's see, we're going to start with this first aspect. First, how do we look at this notion of the tolerated sequence space for a given protein or a given protein-protein interaction, where we then say, well, maybe we can use that to design sequence libraries or to engineer parts families. So you've seen this general scheme of computational protein design. So one of the ways that Elizabeth Humphries in the lab then thought about how to use this to design parts families is by saying, well, really what we'd like to expand this by not looking at one or a few low-energy sequences, but we'd like to have a range of solutions, as many as we can possibly simulate here to generate sequence libraries. In order to do that, what is very likely is what we need to do is that we need to introduce some diversity also in terms of what the structural aspects are of our protein or protein interaction. So we generally use as an input here an ensemble of structures to then generate an ensemble of sequences that we think is consistent with that structure and all its near-native conformational relatives. And we've developed methodology that we published recently in order to generate these ensembles of structures that we've tested in various different ways to represent NMR parameters, for example, or alternative conformations in crystal structures. So then we can generate these sets of sequences. How do we test that these actually represent actual sequences that would be tolerated for a protein or a protein interaction? For this, we used a very extensive, nice data set by Pal et al. That was published a number of years ago, where what they did is they took the extensive 35 amino acid interface between human growth hormone and its receptor and used phage display to screen in total 6 libraries, each of a complexity of 10 to the 10, and sequenced 2,000 different clones to select variants of human growth hormone that fold, but also still are able to bind the receptor. So they had a selection for folding as well as for binding. And they observed at a number of different sequence positions, and again, this comes from 2,000 sequences that they obtained in this screen, that for each of these amino acid positions, there's a number of different other amino acids that are experimentally tolerated to fold and bind the receptor. So we use the procedure that I just described to see whether we can computationally reproduce this by generating a near-native ensemble of both human growth hormone and the interface to the receptor, and then used computational design to predict sets of sequences, and then compiled the variation in this ensemble of the sets of sequences. And we see that, as shown here in 2 different positions in the interface, we find really an encouraging agreement between what's experimentally seen and what we observe in our simulations. So again, I'm showing you 2 cases here. So how would that generally compare over this entire data set of 35 positions and 2,000 sequences? By this metric here, we find that at 16 positions, we actually get defined types of amino acids that are selected more frequently than others. For the other interface positions, these profiles are flat, both experimentally as well as computationally. So then for these 16 positions, we look at how well do we do with our computational library predicting the most frequently observed amino acid at each position. How well do we do in representing or covering these 2,000 sequences? So first, if you concentrate on this axis here, this looks at the % coverage of the predicted library over all the experimental sequences. We see that in many cases, we cover a significant fraction of these observed sequences, except for really here one failure case where we only cover 5% of those sequences. What I'm comparing this to is the best possible case that we call a perfect library. What if at each position, at each of these 16 positions, we pick the same number of amino acids in a library as we computationally predict, but we pretend we know what the answer is, and we predict the most frequently occurring ones. And if you compare these two, the predicted library and the perfect library, you see ideally you would be on this x equals y line here. And in many cases in this interface, we're getting very, very close to this line, showing that we really have a significant coverage that's close to what would be optimal for a library of the same size in this interface. If you're interested in the library sizes here, to screen all 20 amino acids at these 16 positions would not be possible with one library. The computational library now gets into the range where you can do this entire interface. So you would be able to do this entire interface just using one library. Then, is this a trivial prediction or not? We did a number of tests to look at this. Here's one of them, which is this is the plot that you've just seen. But what if you just picked the native amino acid at each of those positions, and you see that you do significantly worse? What if you predicted a naive library where you say, well, if the native amino acid is an isoleucine, we pick things that are similar to it of a certain different amino acid type. And again, this naive library is worse than the computationally predicted library. So then the final method aspect before I go into an example really is how do we look at this idea of interfacing with cellular processes where we both would like to be able to precisely engineer recognition selectivity, but also perhaps try to engineer orthogonal protein-protein interfaces so we can insulate specific interactions from complex crosstalk that might occur in natural occurring systems. So again, here what we'd like to do is to optimize specific proteins to interact with desired partners, but at the same time avoid undesired partners. So one way to think about this is that the scheme of what are the desired and what are the not desired interactions places multiple constraints on a protein-protein interface. And so really what we'd like to have is sequences that are consistent with these multiple different constraints. In order to represent this in the structure-based protein design scheme, one of the ways to do this is to use multiple input constraints, the multiple structures also to then find these multi-constraint solutions that are consistent with a set of structural input constraints. To illustrate this in a little bit more detail, here you would have one protein that has a shared interface with multiple partners. So the multiple constraints here would be defined as trying to engineer this protein to bind to a couple of targets but avoid a competitor, and then get sequences that are consistent with this set of constraints. And we developed a methodology to do this. Not have time to go into detail how we test this, but this work is published. But rather than telling you about the computational tests of this methodology, I'm now going to go into the second part to actually look at an application where what we're trying to do is to selectively engineer a protein-protein interface to try and make it still act on each other but be orthogonal to at least the wild-type counterparts. So again, you've seen this idea of designing specific sets of interactions that work on each other and avoid crosstalk. The simplest way to think about this is the following. What if we have an interface between two proteins, A and B, and we remodel this interface? So now we have two new proteins, A prime and B prime, that specifically act on each other, but at the same time avoid crosstalk with their original wild-type counterparts. So this is really the simplest way of how we can think about this set of orthogonal interactions here. So the model system that we use to demonstrate an orthogonal interaction are rho-type GTPases. Many of you might be familiar with these. They're really central players in the regulation of cell motility, cell polarity, and differentiation. They generally come in two forms: an on state that has GTP bound. This on conformation is responsible to bind to other effector proteins, which are generally then responsible for coordinating a cellular phenotypic output. The off state of the GDPase has GDP bound and generally does not bind these effector proteins. If you activate these GDPases, you can really get dramatic changes in cellular morphology, such as shown here, and this is taken from a paper by Tobias Meyer's lab, where just upon constitutive expression of an active GTPase here, you see these dramatic changes in cellular morphology from this round cell shape into this rugged shape here, where you have these actin protrusions that are called filopodia. So if you get these really dramatic changes upon activation of the GTPase, you can imagine that the switching between these two states is tightly controlled, and that's in fact the case, where there are proteins that are responsible for exchanging this nucleotide, switching the GTPase into its on conformation, and then other proteins that switch it back into its off conformation. And these proteins themselves can then be regulated by other input factors and involved in a variety of complex positive and negative feedback loops. So our goal here was to start with, can we engineer an orthogonal way of activating a specific GDPase by a specific GEF or activator protein, and then show that this would still be able to have a biological output that we can characterize in a cellular system. And so again, we're going about this by looking at the interaction between the GTPase and the GEF and trying to make it orthogonal. So here's the structural detail now of our model system. The GTPase is CDC42, its exchange factor GEF is intersectin. We looked at structural details of this interaction interface. In particular, we modeled one site here. This is the wild-type interface. We remodeled the site to engineer a new interaction here that has an arginine interacting with a glutamic acid. We confirmed this interaction by crystallography, and this is shown here. So we know that these proteins specifically interact with each other. This interaction here is designed, or it's formed as we designed, but it's also a very, very simple change in a sense. It's not remodeling an entire interface, but a relatively localized change in this interface that we started with. But of course, here the goal was not to engineer a completely novel protein interface, but to really see, is this interface now, does it have the desired property that we wanted, meaning is it actually orthogonal? So we tested this first in a very simple in vitro assay. What you do here is you load the GDPase with a fluorescently labeled nucleotide, and you watch the interaction of the exchange factor on the GDPase by exchange of this nucleotide that you can watch as loss in fluorescence over time. So this is a wild-type control. You see a nice loss in fluorescence here. If you make a mutation in just one of the signaling partners, now you see a signal that is not significantly over background, which is just the fluorescence loss of the GDPase alone. If you make the mutation in the other partner, the other non-cognate pair, you observe the same behavior. However, now if you combine both designed cognate proteins, again, you see this exchange of the nucleotide monitored by loss in fluorescence. I should say that this designed interaction here has a significantly weaker affinity than the wild-type interaction, and that is reflected by the fact that to get the same initial rate of exchange here, we need about 10 times more exchange factor. So we have a weaker action of the two proteins on each other, but at least in this particular case, we seem to have made it orthogonal, meaning that it does not cross-react with the original wild-type counterparts in this case. We have other design pairs that are not completely orthogonal, but have wild-type activity or perhaps even faster initial exchange as the wild-type pair. So now, of course, having confirmed this interaction both crystallographically, have done a binding assay, and have done the functional assay, we really wanted to see, can we now characterize this interaction also in cells, meaning that can we introduce this designed pair and see whether it still can drive a certain biological output. And since what we are hoping here is to have an orthogonal interaction, perhaps we can also have control over this orthogonal interaction, perhaps by the action of a small molecule. And then in the future, perhaps take advantage of the fact that we can see whether we can reduce some of the crosstalk, at least use this engineered pair to characterize some of this crosstalk. And so in order to have control over this engineered interaction, we used a system that was published a number of years ago, which uses a small molecule, IRAP, in order to mediate a protein-protein interaction. So the way this works is that we have a CTPA. The GDPA is localized at the membrane. The small molecule can get into cells, mediate this interaction, meaning that it recruits this exchange factor to the membrane that can act to activate the GDPA. So we use that as a small molecule trigger to activate our interaction. And so what I'm showing you now are movies where we have, in fibroblast cells, mammalian cells here, transfected the engineered protein-protein interaction as well as its control here, one of the non-cognate interactions. Let's see when this starts again. So you're starting off with this rounded cell phenotype. You add the small molecule. You see membrane localization of the yellow fluorescence. And then you see these dramatic changes in cellular morphology upon action of the small molecule in the engineered interaction. But here, this is the same experiment. You're adding the small molecule. You see localization to some extent of the yellow fluorescence here, but you don't see these dramatic changes, suggesting that we have an orthogonal interaction that we can control in this particular experiment. We can have these changes in cell morphology, but obviously there's much more to be done now to really build upon the system, see whether we can look at some of these other interfaces and control this in more quantitative ways. So in summary, I've talked about computational design of protein-protein interactions, where some of this detail may be useful and also necessary to interface with cellular processes and perhaps engineer orthogonal interactions that can drive biological output. Challenges are obviously in how predictive is computational protein design, but I've shown you some progress in improving our physical model, particularly in looking at protein-protein flexibility. I've talked about how we use multi-constraint design to try and think about how to optimize molecular parts, in particular proteins, using a mathematical description of multiple constraints, multiple fitness criteria that you might have acting on a protein. The example was to look at specific interactions but avoid other interactions, but you can also imagine that this might be useful to look at the constraints that are imposed on a protein by having to adopt multiple conformations or binding multiple ligands or being soluble or any other description of fitness that you can actually come up with a mathematical description for. Of course, this mathematical description is one of the major challenges, as if you get complex fitness functions, how do you actually optimize efficiently over these multiple criteria? And then finally, I've talked about computational methods to engineer libraries that we are hoping to make consistent, and combine with this methodology, we're hoping to make them consistent with these multiple constraints, to use them directly in parts optimization or in construction of part families that then go back to this concept of tunability and modularity, because then we might be able to design a whole range of modular parts that have varying tunable properties and specifications, because we are exploring a variety of sequences that are consistent with this particular description of fitness criteria. So to thank the people in my lab, you've heard about particularly work from Greg on engineering interactions, from Elizabeth and Dan on computational models. This was helped by Colin Smith, who developed some of the flexible backbone methodology as well. And I stressed two really terrific collaborations, one with Wendell Lim's lab and one with Vigeli Koutsias, and these agencies funded us, and I thank you for your attention.
So the talk is open for questions. Questions?
Thanks. I would like to go back to your sequence tolerance, or the different sequences being compatible with a certain interaction. So traditionally, people have looked at hotspots, and you also have written some program earlier on to actually calculate hotspot energies or make predictions there. So how does that compare? Is that now a new method where you could look at the sequence or the residue tolerance as a measure for the importance of a residue? And is that now going beyond this? I mean, now you have simultaneous replacement of residues, right?
This is simultaneous replacements of residues, and also it's testing the tolerance to residue mutations that are not just to alanine, but also to other amino acids. And so actually in the original paper from Dev Sidhu's lab where they did the library screening, they compared the tolerance profiles that they got experimentally to the hotspot predictions, and there was some agreement, but again they stated there's additional information because they're looking at tolerance not just to alanine mutations. And we find very similar patterns in the computational predictions as well.
Should we just bury the hotspot thing and use this instead?
Yes.
Yeah, question here.
So I was wondering if you could comment just briefly on how computationally intensive the optimization is. What type of resources are required for that?
So in a sense, for designing a protein-protein interface of 20 residues, that can be done on my laptop in, depending on how extensive you model a conformational space, in a few minutes to an hour to overnight. The loop prediction methodology that I showed you, really exploring all these different degrees of freedom, that is quite a lot more computationally expensive. So we generally do this on a cluster of 100 nodes, where we can do the entire data set in about 24 hours. But this is just varying the structural. This is just varying the conformations. We haven't added sequence design to this. So the answer to this is it depends on your level of complexity. You can get fixed backbone, simple predictions very fast, and then obviously you can compute, what the combinatorial complexity would be for adding all these degrees of freedom.
Okay, thank you very much, Tanja.